Thank you. Thank you for this invitation, and thanks everyone for coming. It's nice to see so many of you here. So I'm a professor in vaccine immunology. I'm part of the Immunology and Allergy Unit, the Department of Medicine Solna, so our laboratory is across the street on the hospital side. And I also trained at the Karolinska. I got my PhD here 2001. And apart from working at KI, I have also worked for 7 years at the Vaccine Research Center, which is one of the largest vaccine centers in the world and part of the NIH outside Washington, D.C. And I received my professorship in 2014. So this lecture will be mainly a general introduction to the field of vaccinology. I will discuss some history and some basic concepts, and I'll try to weave in my own research a little bit. But I'm happy to have a deeper discussion afterwards if you have questions or comments. So vaccines have clearly saved a lot of lives, and maybe one of the medical innovations that has saved most lives. Vaccination has led to the eradication and control of several infections. But the vaccine field is constantly evolving, and I was born during the 70s, and I have had rubella and röda hund and mumps, påssjuka, for example, which are vaccines that we are using today in the National Vaccination Plan in Sweden. So with that, I would like to emphasize that you don't have to be 100 years old to have encountered infections that we vaccinate against today. And clearly there are a lot of vaccines that we don't have yet, and they're urgently needed. For example, we don't yet have a fully protective vaccine against malaria, and 200 million people are infected with the malaria parasites every year, and about 500,000 people die. So you can imagine the consequences if we had a vaccine against malaria. About 35 million people live with HIV. There is no fully protective vaccine against TB, and there was a recent outbreak of Ebola and of the Zika virus, for example, and around 4 million people every year die of acute respiratory infections. So there are definitely a lot of things that need to be done in this field of vaccine development. So this is a known painting of the first known vaccination, at least when it was called vaccination. It was around the time when smallpox was a big problem, and people were dying left and right. And Edward Jenner in England realized that milkmaids that spent a lot of time around cows were protected against smallpox. And cows received cowpox that made the cows have blisters, and the cows also transferred that infection to people, but the people got a much milder infection with smaller pustules. But he realized that it was something in this infection that protected the milkmaids. So what he did was that he scraped off the pustule of a milkmaid, and he injected it under the skin of this boy. And the boy developed a mild fever and some pustules, but did not get very sick, and he was protected against smallpox. So after this, Edward Jenner coined the word vaccination, which comes from the Latin word vacca, that means cow. So this was, of course, a big deal in history, but if we should get the facts straight, vaccination as a concept has occurred a long time before Edward Jenner started to do this. It's described for 1,000 years before Edward Jenner did this, what he called vaccination. And in fact, only 30 years before he did this the first time, it was described that the American and British troops fought. This was outside Quebec in Canada. And it was described that the Americans couldn't beat the British troops because they had been variolated with smallpox. They had been injected with a small piece of smallpox blister, again under the skin, and they were protected against smallpox. So the health of the British soldiers were a big deal. And this was when George Washington was leading the American troops, and he realized he couldn't beat them because they had a healthy troop. So he learned from this and started to variolate his own soldiers. But in science, credit goes to the man who convinces the world, not necessarily the man to whom the idea first occurs. And I think most scientists have experienced this in one way or the other, that you have to be persistent in your research. You have to stick with your project, document it well, and then be an advocate for your field. And that's what Edward Jenner did. He did a really good job in convincing the world that vaccination can save a lot of lives. So here is a list of pathogens and diseases, and the year when the pathogen was identified to be the cause of that infectious disease, and then the years when the vaccine was introduced in Sweden, approximately. So we have malaria on top of the list, we have TB. That was the first vaccine that was introduced in Sweden, and this was during the First World War. But it was not safe at the time, and in fact, a lot of young children and infants died of this vaccine. So it has been refined since then. We have several other bacteria here, and we have, for example, one of them is diphtheria, the bacteria that causes diphtheria, and that was the first Nobel Prize in physiology or medicine. Went to the man, Emil von Behring, who discovered the vaccine against diphtheria. And he did that by immunizing pigs and horses, and he realized that they were protected against diphtheria. He could also transfer their serum to other animals, and he found that they were protected against diphtheria. So it was something in the serum that neutralized the bacteria or the toxins. But at that time, it wasn't known that we had antibodies. That was discovered 30 years later. And in the late 40s, a professor here at the Karolinska, Astrid Fagreus, discovered that antibodies are produced by a subset of B cells called the plasma cells. So that, of course, is a huge discovery. And we have one of the animal facilities here on campus is named in the honor of her. Then we have several viruses, and at the bottom of the list here, and second to the bottom, we have the HPV vaccine, which was identified around 81, and the vaccine was first introduced 2006. That is a modern success saga, a quite quick progress from the identification of the pathogen to that we had an approved vaccine. But you may think that 25 years is still a pretty long time. But in vaccine development, this is a success, that you can go so fast to develop a vaccine. So this is a long, long process. So in vaccine development, since vaccines are intended to prevent infections of healthy people, and therefore you don't have a terminally ill patient cohort or something that can first test the drug. It's healthy people and often kids. So there is a very high safety measures, safety precaution when it comes to vaccine development, and also scientific proof that the vaccine is actually protecting against the disease. So at the very bottom we have HIV, and if I would ask you about one major factor why we don't have an HIV vaccine, what do you think I would say then? Mutation, exactly. It's a very, very variable pathogen with millions and millions and millions of different versions. So with the vaccine, it's extremely challenging to come up with something that would cover against all those strains. So if we focused here at the bottom on vaccine variability, you see going from no change to a lot of changes, significant changes over a day. You see that here in the dark red, that's the licensed vaccines we have here listed here. And you see that they have essentially no change. So they are very conserved. Much easier to make a vaccine against them. And here we have HIV. We have influenza here, for example, that we have a new strain every year, so therefore also we have to update the vaccine in order to be protected against that strain that's circulating. So when it comes to HIV and other pathogens that are so variable, it may not be feasible to have a vaccine that induces complete protection, sterilizing protection, like most vaccines we have today. But it may be that you have a vaccine that you'll still get infected, but you will cope with your infection much better. You will have a slower progress to AIDS, and maybe most importantly, you will keep your viral loads so low so you may not transmit them to other people, and that would be a big deal in the world. So there are different types of vaccines, and I would say the best types of vaccines are the live attenuated vaccines, which are based on the live version of the pathogen, but it has been weakened over time. So it should not cause the disease, but you still induce immunity against the vaccine. So listed here we have yellow fever, and that is considered to be the best vaccine we have, the gold standard vaccine. You could have one shot and lifelong immunity. So it's an amazing vaccine, and it was also a Nobel Prize for the discovery and development of the yellow fever vaccine. And we have measles, rubella, varicella, but copper. Those vaccines are also live attenuated, based on live attenuated viruses, and very good vaccines. The problem with infections like HIV, for example, is that you can never have a live attenuated vaccine for HIV. What if it would mutate back to the original version? That would be extremely dangerous. So not even an inactivated complete virus is an option. So the vaccines that are under development today are often based on different types of protein subunits where you take the antigen that you need to have the responses for, and you produce it. Since we have technologies today that allow very precise production of the antigen that you want, and then you can also use different building blocks to put them in a version that stimulates the immune system very well, like a virus-like particle, for example, which the hepatitis B and HPV vaccines are based on. And these are part of, as I said, quite modern vaccines. So this technology is very powerful and seems to stimulate immunity very well. The problem with non-live vaccines is that the immune system may not think that they are dangerous enough, so they won't bother to elicit an immune response. So therefore, with non-live vaccines, you often have to add an adjuvant. And an adjuvant is just something that gives an extra kick to the vaccine for non-live vaccines. And the by far most clinically used adjuvant is alum. It's based on an aluminum salt to which the vaccine adjuvant is absorbed to. It's not more complicated than that. It's been used for 70, 80 years. We don't know exactly how it works, but it's cheap and it's safe, and those are very important aspects. But alum may not be powerful enough for the vaccines that we have left to develop, like an HIV vaccine, for example, or malaria or TB. We have to give something stronger that will push the immune system so that we can get the antibody titers high up and that they stay up high. And we may also have to induce a cellular immune response based on T cells. So therefore, the development of new adjuvant has evolved into a big research area. There are very few that are clinically licensed so far, but there is a lot of work going on. So my prediction is that we will have several in a few years. But one version that is clinically improved is the oil in water emulsion. There are different types, but both Novartis and Glaxo have this adjuvant approved with influenza vaccines. And for those of you who took the swine flu vaccine in 2009, may have discovered that you get pretty sore in your arm after that vaccine, a little bit of a horse bite. And that was an adjuvant effect, because there was an oil and water emulsion, not alum, added to that vaccine. And for those of you who really remember, first the recommendations were 2 shots, and then enough data were out there to say that after 1 shot you get protected. So Sweden bought, I think, 18 million shots, but we only had to use half or less than half. And then there's a bunch of adjuvants that are under development, and I won't go into details here, but toll-like receptor ligands are one popular group of adjuvants where the idea is that you can go in and specifically activate the cells you want to target, if you want to sort of manipulate the immune system to go in one direction or the other. So, as I said, development of a vaccine is a lengthy process, and is often, if not to say always, associated with a lot of disappointments and a lot of failures, a lot of trials and errors. And this is the way it will continue to be. This is a very complex problem to develop vaccines against many of those pathogens we have left that are really challenging. And one problem is also that you may not know what you want to achieve with your vaccine, because nobody has ever been protected against HIV, for example, so you don't have a sort of a benchmark to compare with. It's a chronic infection. And studying people with HIV and their immune responses may not necessarily give you the accurate answer for what you need to achieve with a vaccine in order to be protected. So it is a very complex problem. But I think on the positive side, because as we had on the list before, HIV was identified 83 and 84, and here we stand, we don't have anything close to a licensed HIV vaccine. But both the vaccine development for HIV and for malaria, for example, since they are so large problems in the world, there's also been a push from sponsors, from scientists, from the community to really come up with something. So therefore, so many different approaches have been tested, so many building blocks for vaccines, and therefore, at least the benefits from all this. We have learned a lot, we have come amazingly far, even though there's a long way still to go. But there's a lot also knowledge that have come up about the pathogens, about the immune system, basic components of the immune system, and also methods that are used not only in vaccine development or immunology, but also other areas. So there has been a lot of progress going on, even though there's a lot still to be done. But clear is that we need to have basic research to understand how the immune system reacts to vaccination. We are not where Edward Jenner was, where we can just start to inject stuff into people. We have to know what's going on and exactly put those building blocks together and stimulate that aspect of the immune system in order to achieve what we need to achieve. So my research has been focusing on to understand innate immune mechanism that regulating vaccine response, and by that I mean the first processes that take place when you inject a vaccine. So the first couple of hours, couple of days after you administer a vaccine, when it all starts, how do you give the immune system a push in the direction where you want it to go? So the absolute majority of vaccines work via induction of antibodies. So if there's something you should learn from this lecture, is that how do vaccines work? Well, they induce antibodies and that neutralizes the infection. But to support those B cells, you also need T cells with a helper function, and may also need T cells that can infect, that can kill infected cells. So these components are very important to monitor after you administer a vaccine, but that's sort of what helps you to get protected. What sort of dictates those processes are coming from the dendritic cells, and they're often described as the conductors of the immune system. They tell these cells what to do. And if we had a better understanding how we could provide the dendritic cells, the sort of brains of the immune system, if we can guide them to guide the B and T cell responses, we are in a much better position to develop new vaccines. And for a long time, it's been sort of a black box. What happens when you stick a needle into the muscle? Most vaccines are delivered intramuscularly. And why is that? There are no immune cells really in the muscle in steady state. But that is the delivery technique that's mostly used. But what happens those first days when you start to build up a vaccine response? So during the last years, my research group and a few other research groups have worked on these events, and we have found that after an injection, you induce a mild inflammation at the injection site, and that recruits immune cells into that injection site. It's very specific to that injection site. If you take a muscle biopsy from another site, then you don't have any immune cells, so it's right there. And amongst those cells are dendritic cells. Those cells internalize the vaccine, and this starts immediately after you inject a vaccine. So you can see an effect already after a couple of hours. It usually peaks after 24 to 48 hours, and then it starts to decline. And that's usually when you're sore in the muscle too after an injection. Then these cells migrate to the draining lymph nodes and bring the vaccine with them. The vaccine may also float there by itself. And this transport is very specific from where you vaccinate to the draining lymph nodes. So if you vaccinate in this arm, your migration will occur to the draining lymph nodes in the armpit here, not in the other armpit. And here in the draining lymph node, there is this antigen presentation process. You know, the conductor telling the T cells what to do. And from there, we have the development of a B cell response. And now I'm not going to go into specific details, as I said in the beginning, but we have something called germinal center formation, and that is like a critical structure of a lymph node where B cell responses are born, where they are refined and produced. So from this reaction, we get out memory B cells and plasmablasts. Plasmablasts are short-lived, they produce lots of antibodies, and then most of them die, and some may migrate and take up residence in the bone marrow. So these are components that we can measure to understand the link between here, when you give different kinds of vaccine, to what kind of reactions you get here in the end and development of specific responses. So here is an image of where we have labeled the vaccine, and this is a draining lymph node draining the vaccine site, and this is a lymph node from the other side. And you can see the vaccine only ends up here. And when I started to learn about immunology when I was a student, I had an immunology book that referred to lymph nodes as dating bars for dendritic cells and T cells. Like that's where the action goes on, they check each other out, and if there's a good match, there would be real antigen presentation and it would be T cell development. And there are actually really nice movies about this, scientific movies, from a scientist called Mark Jenkins, if you want to see this antigen presentation process in a lymph node, because T cells and dendritic cells really are dancing around because they need to be a perfect match just for this antigen that is specific for the T cell and T cell receptor. And here is an in vitro culture picture that I took during my graduate studies, where we have a dendritic cell in the middle, and there's an antigen presentation process with lots of T cells, checking out if there is a match. And the identification of dendritic cells and the antigen, their function, was also a Nobel Prize in 2011, given to Ralph Steinman. And this is actually a person that I had the opportunity to interact with. But he died just before the Nobel Prize. I don't know if you remember this, but just before the Nobel Prize was announced. So he never got to know that he actually won the prize. But it was still given to him, even though the rule is that you cannot give it to a deceased person. And here is the last slide I'm going to show you, just to follow up on what's going on with the B cell responses. So usually you vaccinate a couple of times. That's standard. Yellow fever vaccine, remember, one shot, lifelong immunity. That's not the case with many vaccines. So a couple of injections, and what's going on is often that the antibody, you detect the antibodies, they wane, you give the shot again, and you get a better response. And hopefully that response will stay up here. You may have to give a boost again, and hopefully these levels remain high for a long time. So remember, this is the problem with the vaccines under development, where we not know, is this a protective level, or where is the sort of borderline, how long can this wane before it becomes, under detection or under protection. So this is something that's monitored. Sometimes people want to know how are my responses to this particular pathogen? Do I need to get revaccinated? Or of course, it's monitored in vaccine trials. Here is a graph of memory B cells. Remember, the germinal center reaction had memory B cells and plasmablasts. Here comes the plasmablast, short-lived right after the injections. Usually you cannot detect them after the prime, so you have to wait to the boost immunizations. And plasmablasts, as I said, you can detect all this in blood, so only simple blood samples can be used for these analyses. But the real memory is the long-lived plasma cells in the bone marrow. Remember the cells that Astrid Fageus identified, and for that you need a bone marrow sample, but this is really the real reason why you have a long-term memory. So a few take-home messages from this brief lecture. We have different vaccines. I've talked about live and non-live vaccines. And we have an antigen display, and adjuvants offer new opportunities to try to dictate immune responses to where we want to have them. Magnitude and quality. Immune cells infiltrate the site of vaccination, and responses are induced very locally in the draining lymph nodes, so not everywhere in the body, just there. Vaccine-specific antibodies wane over time and reflect the memory cell pool and the levels of long-lived plasma cells in the bone marrow. And now I didn't show so much data, but these are people that have, in my research group, that have contributed to a little bit of the work I showed. And I'll end with this. Courage, and in this case, vaccine development is going from failure to failure without losing enthusiasm. So, thank you.